So, again, we talk about this beautiful chart here that's really laid out nicely for everybody. You're not gonna have to memorize this chart, but you should understand, are you okay?
Yeah, I'm okay. I'm just gonna go. Fine.
You should understand, how these things work for the exam. There'll only be one right answer on your exam, it's all multiple choice and matching. So it's, gonna only have one right answer. So if we think about the immune system, a healthy immune system is involved in some different things. The first thing it's involved in is surveillance. So it's involved in surveillance. What it does is it goes around the body and it makes sure that everything that's in the body belongs there, right? And so if we think about that for a moment, you might think, well, how does that work? Well, I'm going to show you now. The second thing that a healthy immune system does is it recognizes things that are self from non-self. Now, what does that mean? Recognizes things that are self from non-self.
What's in our body and
In
what's
your body,
foreign.
everything is labeled Christy in your body.
Right.
Except your red blood cells. Your red blood cells are labeled different. They have a HLA labeling system. Therefore, we can transfuse blood. But we cannot take a kidney from a doctor and put it in somebody else. The body would not accept a doctor's kidney. You have to take immunosuppressants for the rest of your life, even though she was kind enough to give you a kidney, right? And so it's important. This buddy, when we were growing up, had early onset diabetes, type 1 diabetes, whatever you want to call it. And he needed a new kidney by the time he was 30. His sister gave him one of the kidney, and then he would take care of it all alone. So he ended up all kinds of problems. As you will see with diabetic patients, right? Double amputees, just all kinds of stuff. Because when you have diabetes, not only is your sugar out of control, but you have neuropathy also, and the blood doesn't get to places of the body, and so therefore, when you have a cut and it is a big wound, it doesn't heal very, very nicely. It might take forever to heal. Sometimes, in order to save a person's life, they'll amputate to save. Right? So it's pretty interesting to think about how that works. Once it recognizes self from non-self, then that if it's small enough, the phagocytes will engulf and destroy the organism. If it's too big, like a worm in your muscles, then what it'll do is it'll hold on to it and it'll send out a bunch of messages that basically says, everybody come here and help me destroy this thing. That worm doesn't have a chance. But it's a huge response, and we're going to talk about how that happens. So white blood cells are important for distinguishing self, what's part of you, because everybody's got their own self. Right? Everything in Harlow's body is marked Harlow. Everything in Nikki's body is marked Nikki. Everything in Dez's body is marked Dez. Right? And the white blood cells can evaluate the markers that are on the surface of the cells and determine whether or not it belongs there or doesn't. Okay? And that's because of the major histocompatibility complex. How many people have ever heard of it? Okay, A and P, people. Now don't remember this. Okay, that's not good.
Sorry, what was the question?
Major histocompatibility complex, what is it? There's 4 different classes. I'm going to talk about 2 of them. The other 2 are more physiological and I don't really care about. So when we talk about the major histocompatibility complex, there are 4 classes. I want to talk about 2 of them. So class 1 is DNA that labels or marks, if you will, all cells as self. Okay? So everything in Dez's body is marked Dez because of her DNA that's in every single cell. With the exception of red blood cells, they have a different marking system. Okay? Class 2 are APCs, which are antigen presenting cells. And these are T cytolytic cells that destroy non-self entities and then communicate the non-self to the T4 helper.
Non-cell to what?
Huh?
You said communicate the non-cell to.
The T4 helper.
That says TC.
That stands for T cytolytic, T destroyer, if you will. It's what destroys the cells that are non-self. So can you see this word right here? Jennifer? Jeanette, right here?
Yeah.
That's it. Okay? Good? So, for instance, I am a neutrophil, and that is the bad Lec4 bacteria. So what happens is I'm attracted to that bacterium because it has markers that aren't self. So I go in there and I engulf her, the bacterium. I destroy it completely, and then I'm like, man, I want to keep our helper else. I'm going to help. So then I take a little piece of the protein that I just destroyed completely from that cell, and I stick it on my surface. And then the T4 helper comes and looks at it and says,
It's normal.
It's not. No, well, no.
It thinks it's.
No, it thinks, the T4 helper says this is not, belongs here. So I'm gonna send out a whole bunch of, I'm gonna send out a whole bunch of messages that we call cytokines. And the cytokines are gonna inform the rest of the immune system of the fact that this bacterium was in the blood, or it was in the body. Okay? Isn't that cool? So even that I'm a little bit under the weather, I am probably not contagious anymore, unless you come over and like I sneeze on you or something like that, right? Just like Christy did. But I'm convalesced, and so what's happened is now my T lymphocytes have stopped doing their job, and now what's doing the job is the immunoglobulins, the antibodies that are produced. They're killing whatever's in there, right? So she was kind of sick on Thursday, but now look at her, she looks normal, right?
Hey,
Okay, I'm just
so
saying.
I'm better than I was before.
Okay, good. So, I mean, everybody's got a different physiology, everybody's got a different immunology. So it doesn't take me very long to get over stuff, right? Some people take a lot longer. Anybody have any questions about the major histocompatibility complex? I'm sure you're going to see questions about it. Okay? So, again, here's the white blood cell. Here's some cells. They have the self markers, so when the white blood cells come in contact with them, they just leave alone because they're marked correctly. And the body's going to be taken care of by the immune system, right? So those cells that are marked part of us, not going to be hurt. Something comes in and has different markers, then the white blood cells will completely engulf them and destroy them. Okay? And then whatever's left over gets communicated to the T4 helper. The T4 helper is the most important part of the entire immune system because it is the only one, the only cell that can turn on the entire immune system. There are parts that can turn on parts, but they can't turn on the whole thing. The only thing that can turn on the whole immune system is the T4 helper. Okay? Anybody have questions? And that's why HIV and AIDS is such a terrible diabolical disease because HIV targets the one cell that can turn on the immune system. That's a T4 helper. And once your T4 helpers are below 200, you have no immune system. And even if everything else is there, the T4 helper does not initiate the immunology, the immune system. And so a lot of times people with who have full-blown AIDS die from some disease that doesn't really kill people, like pedicillosis or aspergillosis or cryptosporidiosis or toxoplasmosis. There's a bunch of them, right? But these are things that normally don't hurt people because we have a good immune system. But when you don't have a good immune system, there's no fighting. Yeah.
You said below 200 or 400?
Below 200.
So AIDS itself doesn't really kill you. It's because the immune system is dropped and your
That's
body
correct.
can't protect
You do
it.
not die from AIDS.
Got
You
it.
die from some opportunistic disease or cancer that develops because you have no immune system. That's correct. People don't die of AIDS. They die from something else.
But they like to say you died from AIDS.
Huh?
People do say, oh, they died from AIDS.
Yeah, but they don't know what they're talking about.
They do because of AIDS.
Right. That would be a better way to say it. And to be perfectly truthful, you could say, my friend died of complications because they had AIDS and they died from
A weak immune system.
an infectious agent that overtook them because of a weak immune system. Or cancer. Kaposi sarcoma is a cancer that we never saw in anybody except Italian men in their late 50s. And in the late 80s and early 90s, we started to see it in American males who were in their teens or early 20s. That is a big shift. Big shift. Okay? Anybody have questions? Okay. So, it's about the beta lymphocytes and the T lymphocytes, right? And there's two types of immune responses. There's the cell-mediated, that's the role of the T lymphocytes, and the humoral immunity, which is the role of the beta lymphocytes. And so, if we think about it, the term antigen is synonymous with non-self. So, if there's an antigen in your body, you have a non-self entity in your body, and that non-self entity can be anything. It can be any infectious agent, but it can also be pollen. It can be a kidney from somebody else. It can be that you got a blood transfusion and the blood they didn't do a good job of cross-typing and matching, and your immune system is now wreaking havoc on that blood. It's destroying it because it doesn't belong there. It is amazing how this immune system works. So antibodies are a group of proteins that are produced against a non-self entity. And every single non-self entity that gets in our body, whether because we have an infection or because we've taken a vaccine, the immune system doesn't recognize the difference. It's all not good, and therefore it produces antibodies against whatever gets into our body that doesn't belong there. Right? That's why vaccines are so cool, because you take a little bit of non-self entity, usually in pieces, not even the whole bug or the whole infectious agent, but the immune system will recognize it as non-self, make immunoglobulins against it, and then you're protected.
So it's almost like, for instance, like Botox, people can create antibodies with it.
People can what?
Create antibodies with Botox.
Because it's a protein. Yeah. And that causes inflammation and all kinds of craziness. That's why you mostly give it superficially, because there's less body in the superficial layers of the body than there are in the tissues. So we have two types of immune systems or immune responses. Cell mediated, that means that the T lymphocytes help the immune system, and the humoral, that means that the beta lymphocytes release plasma cells, and the plasma cells produce immunoglobulins. Okay?
Can you say that one more time?
Yeah,
The
here it is right here.
Oh,
So if you think about it, the humoral immunity produces antibodies, and the cell mediated, it's the T lymphocytes itself that does the destruction. Okay?
Can you go back to the other slide?
Yeah, the first one, the two, of humoral, okay.
Okay, where does humoral come from? The humerus, larger bone of the body, and the reason that is because all of these immune cells start off in the bone marrow. That's where they're made. As a matter of fact, here is the bone marrow, and here is the stem cell. And now it's a lymphocyte, and it goes to the thymus, and in the thymus, it becomes specialized. I like to say it becomes differentiated because it now becomes a T lymphocyte. Thymus derived. Thymus derived. Therefore, it's a T lymphocyte. And then the T lymphocyte can either be regulatory, they can be cytolytic, or they can be helpers. Okay. On the other side, in the bone marrow, we used to think it was around the bursa, so therefore it got the name B lymphocytes because it was bursa derived. We now know that's not true. What is the bursa? It's the area around the joints, right? We now know it's not the bursa. We now know it's the bone marrow where they become differentiated. So in the bone marrow, these stem cells become beta lymphocytes, and then they themselves secrete plasma cells, which will then secrete immunoglobulins. We just got lucky that bursa and bone marrow started with B's. We'd have to change everything.
Thank God.
Yes.
That's his wife.
Yes.
She doesn't remember.
Why?
Exactly.
Okay, what happened?
And the ringtone
What
sounded
happened?
lovely.
It did.
Okay, what happened? Which examiner? Oh, well, that's not good. Okay, well, that's okay. Well, I'll just have to talk to him and have to take it again.
It's like, or the immune response?
Yeah.
Oh, I just forgot.
Yeah, he can take it again.
Start at the
Okay,
very bottom
what am I?
of the PowerPoint.
Bye.
Immunology?
Or is it the immune response, the second one? under
What PowerPoint
determinants?
are you on?
What?
Immunology.
What'd she say?
Immune response one?
Immune response. It's
Okay,
the
okay, yeah.
Yes.
And then when you open it, it says.
What are we talking about?
Well, you
You
mentioned-
changed-
We thought you had added on extra slides, and I've been sitting here going, okay, I guess they'll add it later, not realizing we were on the next PowerPoint.
I know that
Girl,
happened to me.
I'm moving fast.
I'm like writing it on the other PowerPoint.
I know.
Like, where's this coming
It's
from?
Some of those slides are in the last.
Yeah, I know.
PowerPoint. That's why they're probably not.
Oh,
Because
that's- yeah.
sometimes I don't open up the new one before I finish, because
Got it.
depending on time, but I have a lot of time, so I just open up a new one.
Got
So
it.
that's why we thought.
Ah, okay.
Yeah, and then sometimes we'll start at the bottom of a PowerPoint and then jump around, so
I don't jump around, I go step by
up.
step.
Nope.
Okay, here we go. So, I say that, I'm gonna jump around. So, the T lymphocytes either become T helper, T cytolytic, or T suppressor. Now, it's much more complicated than this, but I'm gonna talk to you this way, because at this level, you just have to understand what they're gonna do. You don't have to know them by T4s or T8s or T25s or whatever, you just have to know what they do, okay? So if you think about this, the most important of all of the T lymphocytes is the T helper. The T helper is the one cell in the entire body that can turn on the entire immune system. The only one, okay?
This
And-
is T4?
T4, also known as a CD4, they're the same, okay? So without this thing, the immune system doesn't work. All with me? Okay, so the other types of that we're gonna talk about here, we're not gonna give them numbers and initials. We're gonna say T helper is involved in recognition. Now, how does that work? How does the T helper recognize self from non-self? How does that work? How does it say, oh, you belong here, and how does it say, oh, no, you don't belong?
Maybe because the shape.
No.
Label.
Huh?
It's labeled?
Label?
The labeling, yeah. What do we call the labeling?
I was gonna say DNA.
You started out so good, Jenny, and then you just stopped.
Well, you
Is it
asked me a
DNA?
question.
Huh?
I was gonna say DNA. Major histo.
What was it? Major histo.
Compatibility complex. Every single cell in your body is labeled self. So the T4 helper and all the other cells know what to look for. They're looking for you all, and if your cells are there, they're gonna leave them alone. But if something else is there that doesn't have the markers, they're gonna be destroyed. Isn't that cool? Wow. And so now, if you, I'm not gonna show you because it's kind of gross, it's not gross to me, I'm like, oh, that's great, look at that. My mucus is yellowish green, and the reason
Oh,
it's
so was
yellowish
mine.
green is because I am now convalescing, I'm getting better, and all the IgA and the macrophages are all doing the work, they're cleaning up the virus that is in there. It's all beat up, it's just, they're destroyed. Thanks, Christy.
I know, it's not fun.
He's nice.
No, no,
I
no.
know.
Every once in a while,
Right.
it's okay. I always try to update my immune system at least once a semester.
Yeah, I'm glad,
And
you're
here's the
welcome. I'm glad I could help.
You noticed, I don't know if you noticed in class, I said, Christy, come over here, and she's like, I don't want to get anybody sick. I'm like, I'm not afraid of you, come over here. Well, I knew what was gonna happen, I knew I was gonna get sick. So it was a plan, mostly because I wanted to make fun of her in class.
Yeah, cause I know.
It's fine, yeah. It could have been you
Got
too.
thick skin.
Oh, you don't get sick.
Oh, I better knock on wood. Better knock on something.
I bet if you rub these skunkies on your nose, I bet you'll get sick.
Start talking.
Okay, so I'm not gonna expect you to know how they stain, that's the anatomy and physiology thing. But I do need you to know that neutrophils and eosinophils are capable of phagocytizing pathogens. Okay? Now, if we think about that for one thing, neutrophils and eosinophils are also capable of diapedesis. Who knows what diapedesis is? What is it, Jack?
It's it's like the cell moving like that.
The white blood
blood
cells
cells
can move
can-
out of the circulatory system and into the tissues of the body.
Mhm.
Why is that important?
Say that again?
The white blood cells can move out of the circulatory system and into the tissues of the body. Why is that important, Bree?
I don't know. I was writing it down.
You do know.
I can't do multiple things at
Yes, you
once.
can.
You said it moves to the skin?
Huh?
Tissues. Oh, tissues. Tissues of the body.
You saw the tissues on your mind.
Why is it important? Atate. Why is it important that white blood cells move out of the circulatory system and into the tissues of the body?
Fight off
To
infections.
fight off the infection. Because if you have an infection of the lungs, and the white blood cells are only in the arteries or the venous system, they're not gonna do you any good. They have to get into the lungs to destroy whatever's there. Isn't that cool? It's
Dude, I learned more with you than I did with my AP teachers.
What's that?
I said I learned more with you than I did with my AP teachers.
So it's interesting because the A&P people, they voted without the micro people being there. And guess what they voted?
Huh.
We vote that we don't talk about immunology, we'll let the micro people talk about it. They voted that and didn't tell us anything. Cut that out. Okay, now listen. So here's the other really interesting thing I want you to know. Eosinophils show up when you have allergic reactions and if you have a parasitic worm. Isn't that interesting?
Show up on what?
When you have allergies or a parasitic worm infection. Okay? But the neutrophils show up when you have bacterial infections. Always. Okay, so now look at here.
Dude,
What's going on here? This is not a human. I have a hard time getting really good slides from human. This is a dog. But dogs work just fine because they kind of work the same way we do. What's going on here?
Eosinophils.
No, not eosinophils. Neutrophils. Why do you say neutrophils? I agree. Why do you say that?
Because they're green.
Well, what is the gram specificity and the morphology of those cells right there? GPC, gram-positive toxic. Well, we just said that neutrophils show up when GPCs are there. Look at these neutrophils. They have engulfed those cells. They're doing a really good job. This dog's immune system is protecting. It happens the same way in us. Isn't that cool? Which is what we call phagocytosis. You need to know the parts of phagocytosis. Luckily for me, I have a really good slide of it here. So phagocytosis, the way I remembered it in 9th grade was to remember cake.
I almost said that.
Huh?
I literally almost said that.
You should have said it.
I was like, cake.
Cake. C for chemotaxis, A for adherence or attachment, I for ingestion, K for killing, and E for elimination. Cake. It's a mnemonic. It's a good one. What's that? No, not tres. Although you can bring tres leches anytime you want.
I love
Yeah.
that.
Okay, so check it out. So first of all, look at how big this yeast cell is. With this white blood cell, it says, I don't care how big you are, I'm going to kill you. And it's beginning to engulf it. Look at that thing. They're about the same size. But the white blood cell is like, not going to be a problem. I'm going to get you. So here is the process, right? So here you have your macrophage, your phagocyte. Here you have your bacteria. And the macrophage is attracted to the bacteria because of chemotaxis. Chemotaxis is the attraction to chemicals. In this case, it's attraction to the non-self markers on the surface of those particular bacterial cells. So it's going to be attracted to them, it attaches to them, and then it brings them in a vesicle that we call a phagosome. It's called a phagosome because through endocytosis, those particular cells have been brought into the phagocyte. Yeah? But that's not all. There's lysosomes. You remember lysosomes? We talked about them. Lysosomes have digestive enzymes in it. Now one of those lysosomes is going to fuse with the phagosome. It's going to become a phagolysosome, and the digestive enzymes are going to completely destroy the non-self entity. Completely. And then whatever's left over, it's just going to be eliminated into the blood, and the kidneys will filter it out, and you'll pee out the residue. Are you with
That's
me?
the elimination.
Well, the elimination is just eliminated from the cell, but I guess you could say it's eliminated from the body through the urine also. So, you should never have protein in your urine, right? But if you've had trauma, right? I don't know, you're playing football and somebody tackled you and you got hurt, or if you've had an infection, then you're going to see some protein in the urine for a little while until the body cleanses it out. But normally, you should not see any protein in the urine. Okay? What kind of what? Well, you wouldn't be able to see it, but you could test for it.
I think for women, isn't it like right after periods, right?
Well, that's for a different reason. So, when you collect a urine specimen from a young lady, you should always ask, are you undergoing menensis? Because if they are, you have to treat the urine separate. You have to treat it differently, right? You can't count, like if you have 80 red blood cells per high-powered field, you can't say that that's abnormal because they're undergoing menensis. So you have to understand those things, right?
That's why they always ask women for a urine sample.
Well, they asked men for one too. I just gave one on Friday, and that lady who stuck me. Look what she did to me. She gave me a big old bruise. I'm like,
Rookie.
I'm gonna write a letter.
Right?
Yeah, mine, I got my vaccines and she was trying to inject me HPV on the back of my
Well,
arm.
that'll work. It can be anywhere.
It was hard to poke through though, and I have a ball ever since.
They could have given it to you on your foot.
Ouch.
But she said it's supposed to be intramuscular because it was hurting.
Intramuscular, yeah. Die?
Yeah, but she was giving it to me like fat.
You don't have that much fat.
I
right?
do have arm fat.
Okay. I'm not gonna argue.
Okay.
Any questions? Alright, here we go. So we talked about major histocompatibility. Let's review. Class 1 of MHC is what? DNA. It labels everything south. Class 2 are what?
TC.
APCs. May I introduce you to the way the process works? Boom. Here they are. There's your dendritic cell. This is your APC. It's attracted to that bacterium. It brings it in. You see it's all dissolved. But you have a little bit of protein left over. And it sticks it on the surface of the cell. And it communicates with the T4 helper. The T4 helper says, oh, this doesn't belong here. Sends out cytokines all over the place. And the cytokines tell the rest of the immune system, be on the lookout for this thing. It doesn't belong here. If you see it, destroy it immediately. Right? So now all the cytolytic cells are looking for it, and the beta lymphocytes are making immunoglobulins, antibodies against that specific non-self-active. Isn't that just cool?
We need to know this whole process.
What's that?
We need to know that whole process.
You need another one.
Do we need to know that whole process?
Let me think about it. Yes. It's gonna be multiple choice. It's gonna be right there. All you gotta do is pick the right answer. So, here's the really cool thing. Here's your antigen presenting cell. It's presenting the antigen to the T4 helper. Now the T4 helper can send signals. It can send it to the beta lymphocytes, which then will secrete immunoglobulins. It can send it to release a whole bunch of macrophages, right? Monocytes first, and then you get macrophages. Or it can send it to the cytolytic cells that says you all need to be on the lookout, and they become mature, and then they will destroy anything they come in contact that is labeled that particular non-self protein. Okay? So this is elephantiasis. How many people have heard of it?
I have.
What is it?
It's when the body is inflamed and it can't like go through it can't go through the lymphatic system.
It can't what?
It's like something like it that can't go through the lymphatic system.
Correct.
Like
What causes it?
I'm guessing blockage.
Okay, good. But what is the cause of the blockage? It can be two things, right? It can be an overproduction of fluid by the body, which we call ascites, or it can be microbe-driven. And in this case, this is Wushia bancrofti. This is a little bitty filarial worm. It's a parasite that actually gets in here and it clogs up all the lymphatic system, so you develop this huge amount of inflammation. We do not have this organism in the States. It's in Asia, right?
But I feel like I've seen somebody like,
on
a train, like not on a train, but like I've seen somebody in public like with that.
Oh, well, okay. It's probably just edema that's due to malfiltration by the kidneys. That's very common. You'll see people with very
Red and kind of. Yeah.
and swollen, especially
Diabetics.
lower limbs and feet.
Mm-hmm.
Can we get it drained?
You can get it drained, but it's very costly.
Is that also, and that kind of coincides with like gout too?
That's different.
Okay.
The gout's different. It's
Right.
the buildup of uric acid. That's different.
Okay.
But you can get it drained. It's going to be through dialysis, right?
Mhm.
Or they can give you this stuff called Lasix. I don't know if you've heard of it, but it will help you eliminate some of the excess fluid. It doesn't work right away. You're going to be on it for a while. Did you have a question or?
Well, so with edema, something like that, would that be both legs? To like both of them would have a similar kind of issue, or can it just
Like both legs?
So like here you can see it's obviously like that's the normal leg, and then this is not
Yeah,
normal.
yeah.
But with something like what you're talking about,
Yes, with diabetic patients who have that type of improper filtration of the fluids that are produced by the body, you usually see it in both legs. That is true. Yeah. And it looks painful because if they're wearing sandals, the straps of the sandals, they're putting so much pressure on those tissues that it looks like it's gonna just break. I mean, it's very sad.
What causes it to get like the crustation? Like that's what so the redness, that you see in like you said, it's in lower extremities, but where it's red, like sometimes it looks really dry and just kind of almost, and it does, like kind of flaky.
So that is neuropathy. That is not getting proper blood supply.
Okay.
And so you're having inflammation, but the inflammation is actually causing the skin to like expand, if you will,
Okay.
which is causing the redness.
Yeah, okay.
And it's very itchy too.
Yeah.
Why is this not here in the US?
This one,
Yeah, you
be
said.
lucky, girl. We don't want.
I don't have that
I don't want this guy
parasite.
here.
No, I just wanna know.
It's
Because they're not
regional
indigenous.
because there's a black fly that actually
Why?
there's a fly that actually carries it, and we don't have that fly here.
Got it.
We got all the other ones.
And Bree, don't go out there and pick up some of those flies and bring them back. We don't want this thing here. Okay?
So if
And
a
it's an
person
Asian-
has it and they come to the States, it's not transmitted.
Except we don't have the vector. So it's gonna be really hard for it to be
And-
spread because we don't have the vector.
And the vector.
Now, if you do a blood transfusion, that's a whole different ballgame. But we're really good at looking at somebody and saying, I don't think so. You don't look exactly up to snuff right now. Come back in 3 months, you know. And I say that in jest, but there is a lot of evaluation by the phlebotomist prior to actually asking the questions, right? If they're looking at you and you look a little bit skinny and a little bit pale, they're like, nah, I don't think so. Yeah. And they'll say things real nicely like, I thought you only weigh 100 pounds, you have to weigh 110 pounds. And you're like, but I weighed myself this morning, I weighed 120. Our scale says you only weigh 100.
That's nice.
Yeah.
Right.
But some people will say, I can't believe it. Let me weigh myself. And you're like, no, it's okay. You've already been asked to not donate to me. Because when I was working for J&J had this, like they had a van, like a big old truck bus, come over and people would go donate blood, right? But some people didn't want to, and then some people were pressuring people to do that, right? And so in order to do that, they would go over there and then they would answer yes to a question that they never should answer yes to, right? Like, for instance, have you been exposed or been tested for HIV and tested positive? And they would just say yeah, because it's all confidential, they would just say yes, just so they didn't have to donate blood. But then that went on their record. Yeah, you can't lie to the medical professionals, it ends up on the record. Are you with me? Yeah. Years ago, my, turn it off.